Okay, good morning, everyone. All three of you. Wow. Look at the attendance. It's falling down dramatically as we get close to the end of the semester. I think people are starting to feel very stressed out. So thank you for showing up this morning. I hope you're doing well. Let me mention to you that the final exam schedule has finally been released by the administration. And so I can tell you that at this time, your final exam in this course will in fact be Monday, May 11th. Okay, your final exam is Monday, May 11th. I will indicate that in my email to you this afternoon so that you'll have that documented. But once again, the final exam will be on Monday, May 11th. Okay, do you have any questions at this time about the course, about how the semester will end, anything that you would like to ask about? Okay, if you can hear me okay, would you please type yes into the chat window? All right, great, thank you so much. All right, so what we're going to do at this point is finish up chapter 12 today. I would love to have another week with you on this content, but we just don't have it. So we're going to do our best to get to an action potential today, the physiology of it. And then next week we'll be starting the next chapter, chapter 15. Okay, so do your best to stay focused and stay up with your reading. I know it's hard right now because there's plenty of other distractions. Okay, so I'm going to go to our Microsoft Word document and start taking some additional notes about the action potential. So bear with me one moment. Okay, so this is Biology 121, lecture on chapter 12, part 3. Today's date is the 22nd of April. Okay. All right, gang. So as I alluded to in my past email, I want to start today with a video that gives you an illustration, a cartoon illustration of what an electrical signal in the nervous system really is. Okay, so I'm going to begin here by showing you this video online, and then we can talk about it, and we're going to step through several important aspects of the physiology that the video is going to talk about. So bear with me. I'm going to bring up a YouTube video on the electrical signals in the nervous system. Okay, here we go. All right. So try to stay focused. Try to really think about what this video is showing you. And of course, I have sent the link out for it. I'll send it again and you can watch it at home. Here we go.
How does the pain you experience when you burn your hand result so quickly in an action by your muscles? Many animals respond to environmental stimuli using specialized cells called neurons. A stimulus is detected by sensory receptors, and the body responds through motor effectors. These cells working together allow you to respond very quickly to threats. When you touch something hot, heat receptors of a sensory neuron detect the stimuli and send the information of heat to an interneuron in your central nervous system. From there, a motor neuron sends a response from your central nervous system to the skeletal muscles in your arm, causing them to contract and pull your hand away. The fundamental process of neural transmission that underlies this action occurs in all neurons of the body. Neurons transmit this information through changes in the electrical potential of the membrane by the movement of ions across the membrane. An electrochemical gradient governs the movement of these ions, resulting in an electrical impulse. The resting membrane potential in a neuron, when the cell is not firing an impulse, is established by the unequal distribution of sodium ions outside of the cell and potassium ions inside the cell, making the outside of the cell more positively charged compared to the inside. The electrochemical gradient is established and maintained by an enzyme called sodium-potassium ATPase. When a neuron is stimulated, sodium ion channels open and sodium ions flow into the cell. This leads to a change in the electrical potential across the membrane called depolarization. The depolarizing electrical potential travels down the dendrites and over the cell body. Multiple electrical potentials will combine at the axon hillock in a process called summation. If the depolarization is large enough, an action potential is triggered. Action potentials are all or none electrical impulses that maintain their amplitude and strength down the length of the axon. The action potential travels down the axon when the depolarization of an area of membrane causes adjacent voltage-gated sodium ion channels to open. The influx of sodium ions results in membrane depolarization along the membrane. After a short delay, potassium ion channels open and potassium ions flow out, repolarizing the membrane. For the neuron to fire again, the resting membrane potential needs to be re-established. Sodium-potassium ATPase is used to move sodium and potassium ions against their concentration gradients, re-establishing the resting membrane potential. As the action potential moves down the axon, ions are diffusing only a short distance, allowing the signal to move quickly. At the axon terminal, the electrical impulse passes to another cell at a cellular connection called a synapse. The space between the presynaptic neuron and a postsynaptic cell is called the synaptic cleft. The presynaptic neuron contains signal molecules called neurotransmitters that are packaged inside vesicles. When an action potential reaches the end of a neuron, neurotransmitters are released by exocytosis from the neuron into the synaptic cleft. Neurotransmitters bind to the adjacent cell at receptor sites attached to ion channels. The channels open, allowing the movement of ions into or out of the effector cell, which alters its membrane potential, thereby transmitting the signal from the neuron to the effector cell. Because nerve impulses move very rapidly down the axon of a neuron and move from cell to cell across synapses, you react quickly to a stimulus, like burning your finger.
Okay. Okay. So folks, after you've watched that video, do you have any questions that you'd like to ask at this time before we get into the nitty-gritty details of things? Anything on your mind? Okay. All right. Everybody's silent. So let's begin. Okay. What I'm going to start talking about is content on pages 418 and 419 of your textbook. The very first thing we need to talk about a little bit is the resting membrane potential of the neuron. Okay, so a couple things here to keep in mind. If you take a look at figure 1213, that would probably be a good visualization for yourself at this time. Figure 1213, what I want to point out is that you have a variety of ions in the extracellular fluid outside the neuron, outside the cell. You also have a variety of ions inside the cytoplasm of the cell, inside the cell, internal to the plasma membrane. On average, you have roughly equivalent numbers of positive and negative ions outside the cell in the extracellular fluid, and roughly equal amounts of positive and negative ions inside the cell in the cytoplasm. And the consequence of that is that you appreciate that on average, the extracellular fluid is neutral. And on average, the internal cytoplasm of a cell is neutral. Okay. So on average, the cytoplasm inside a cell and the extracellular fluid outside the cell have an even mixture of positive and negative charged ions and molecules. Have an even mixture of positive and negatively charged ions and molecules. Okay? So the take-home message is that the cytoplasm and extracellular fluid are the cytoplasm and extracellular fluid have a neutral charge. That is to say, they're relatively uncharged environments. Okay? Now, that circumstance of having a neutral environment actually is not maintained at the most intimate surfaces of the plasma membrane itself. If you look very, very carefully at the plasma membrane in figure 1213, you'll notice that the outer surface of the plasma membrane surface that is immediately exposed to the extracellular fluid, has a bunch of positively charged ions there. And in the same vein, in the same way of thinking, the most immediate surface interior to the plasma membrane that is most interiorly exposed to the cytoplasm has negative charge. Okay, so I have a question on the chat window. Yes, Ali, the extracellular fluid is outside the neuron. It's the fluid outside the neuron. Okay? Any other questions? Okay, let's keep going here. Okay, so along the inner surface of the plasma membrane, negative ions tend to, well, let's put it this way, negative charge tends to accumulate and along the exterior surface of the plasma membrane, positive charge tends to accumulate. And there's a reason for this, okay? So the first question is why? The answer for this is because of the leak channels that are embedded in the plasma membrane. If you look carefully at figure 1213, you notice embedded in the phospholipid bilayer, there is a blue-colored potassium leak channel, blue-colored protein, if you will, and there is an orange-colored sodium leak channel, an orange-colored protein, okay? These channels are constantly moving ions back and forth across the plasma membrane. But as I mentioned at the end of my lecture last time, potassium leak channels are leakier than the sodium leak channels. That means on average, for every 1 molecule of sodium that you bring in through the leak channel, you bring 2, 3, or 4 potassium ions outside the cell. So that means we have more positive ions leaving the cell than we have entering the cell. That means there's an opportunity for more positive charge to accumulate on the exterior surface of the plasma membrane. Okay, so remember, the answer to the why question, why this happens, that we have a negative charge on the inner surface and a positive charge on the outer surface, is because leak channels are constantly moving ions back and forth across the plasma membrane. On average, more positive ions are leaving the cell than sodium ions enter the cell. Okay, on average, more potassium ions leave the cell than sodium ions enter the cell. This is the reason that more positive charge builds up on the exterior surface, and more reason why an absence of positive charge or a negative characteristic, a negative charge, accumulates on the interior surface of the plasma membrane. Okay, it's not that, the interior surface, it's not that it's truly negatively charged in the fact that there's a bunch of negative ions. The negative characteristic is really the absence of positive charge because the potassium ions are leaving, okay? So while a negative charge, which is really the absence of a positive charge, builds up on the interior surface of the plasma membrane. Okay. All right, so just a basic question coming through the chat window here. Okay, is this making sense so far? Are you understanding what the video is showing, what the picture in your book showing, and what I'm trying to communicate through the computer? Is this making sense, ladies? All right, very good. So remember, the potassium channels are leakier than the sodium channels, and on average, in any given section of plasma membrane, typically you will find more potassium leak channels than sodium leak channels. That means, on average, you have more positive potassium leaving the cell than you have positive sodium entering the cell. It's for that reason that you have more positive charge on the exterior surface and more negative charge on the interior surface of the plasma membrane of the neuron. Okay. Now, because of what we just explained and written down in bullet point number 2 on the notes, it's important that you appreciate that because of this distribution of positive charge on the outside and negative charge on the inside, that means there's a separation of charge. And the separation of charge, you should recall, we used as a term when we talked about water. Water has a separation of charge, right? And we often called water a polar substance. Well, in such a case, the resting membrane potential of a neuron is polar. In other words, the membrane is polarized while it is at rest. Okay, we can apply the word polarized to the plasma membrane of the resting neuron. In other words, the plasma membrane of a resting neuron is polarized. Okay, you might ask yourself, well, how polarized is it? what is the actual difference? What is the difference in electrical charge across the membrane of a resting neuron? And can you answer that for me? If you know what the resting membrane potential of the neuron is, please type that into the chat window. Can you answer the question, what is the resting membrane potential? We need to put a number on it. Can you tell me that? Okay. It's not 70, Pam, because your 70 is a positive number. Is that what you really wanted to write? There you go. Okay, it's minus 70. Exactly. In other words, if we hooked up a voltmeter, which don't worry about it, I mean, maybe you have some experience with electronics, but if you hooked up a voltmeter and you hooked up an electrode through the plasma membrane as they're showing you in figure 12.12, picture B there, that instrument would in fact measure minus 70 millivolts. That is the resting membrane potential. Okay, minus 70 millivolts, okay? All right. So I just want to be sure you appreciate the resting membrane potential of a typical neuron is minus 70 millivolts. Okay, you don't have to worry about, a volt is the unit of measurement that is used by scientists to measure the difference in electrical energy across a surface, like across a membrane, like across the plasma membrane of a cell. Okay, all right. Any questions? All right, now let's talk about the factors that influence this resting membrane potential. There are in fact three things that need to be said. First off, there's an unequal distribution of ions between the extracellular surface of the plasma membrane and the internal surface of the plasma membrane, okay? There's an unequal distribution of ions in the extracellular fluid and the cytosol, okay? Going a little bit deeper here, the extracellular fluid, the ECF, right? The extracellular fluid, the ECF, is abundant in sodium and chloride ions. And the cytosol, the cytoplasm, the interior of the cell, is abundant in potassium ions and phosphate anions, which would be PO4 with a -3 charge. Okay, PO4 with a -3 charge. Okay, so that unequal distribution helps create the polarized effect of the membrane potential. In addition to that, we realize that most of the anions that are inside the cell, most of the negatively charged ions, are not able to actually leave the cell. And that's because many of them are too large to actually diffuse through the plasma membrane, while other negatively charged ions actually end up attaching to other molecules in the cell. And because they're attached to the larger molecule, they cannot get through the plasma membrane. Okay, phosphate anions, the negatively charged ions, are part of larger molecules that are too large to diffuse through the plasma membrane. And thirdly, the third factor that influences the membrane potential is what is referred to as the electrogenic nature of the so-called sodium and potassium ATPase pumps embedded in the membrane. Let me write that down, then I'll talk about it a little bit. Okay. Okay. So that term electrogenic, let me get it spelled right here, electrogenic. There we go. Okay, electrogenic is simply a fancy way of saying that the sodium potassium ATPase pumps, that's what they are. They're just pumps. In other words, because they're pumps, they have the ability to change the potential of the membrane. Electrogenic simply means that these things are ion pumps and that they can change the potential of the membrane. Electrogenic is a fancy way of saying that the sodium potassium ATPases are ion pumps that can move ions in a way that influences the charge difference across the plasma membrane. Okay, electrogenic is a fancy way of saying that the sodium potassium ATPases are ion pumps that can move ions in a way that influences and alters the charge difference across the plasma membrane. That's all that that word is implying. Okay, so you've probably read it already in your personal study time. It's important that you appreciate that these sodium potassium ATPases are able to actually pump out 3 sodium ions that have leaked into the cell through the leak channels. Okay, sodium potassium ATPases actively, meaning that if it's active, right, they're going to require ATP, and that's hinted at in figure 1213. You see ATP is being consumed there on figure 1213. Sodium potassium ATPases actively pump out 3 sodium ions that have leaked into the cell while pumping 2 potassium ions that have leaked out of the cell back into the cell. Okay, so let's highlight some important ideas here. The sodium potassium pumps pump out the sodium. They pump out the sodium. The potassium pumps it back into the cell. Okay. All right. In other words, the sodium potassium ATPases are moving the sodium and potassium ions in the opposite directions that the leak channels would move the same ions. Okay. Notice that the sodium potassium pumps move the ions in the opposite direction as the leak channels. Okay, so there's a way to move the sodium back and forth across the membrane. You just have to use the appropriate channel. If we want to get sodium into the cell, we use a leak channel. If we want to get sodium out of the cell, we use the sodium potassium ATPase pumps. If we want to get potassium out of the cell, we use a potassium leak channel. If we want to get potassium back into the cell, we use the sodium potassium ATPase pumps. Okay. Folks, do you have any questions at this time? All right. Okay. So now what we're going to do, now we've established why. Okay. I hope we have explained, I have explained, the book and animation has explained why there is a difference in electrical charge on the outside surface versus the inside surface of the plasma membrane of the neuron. Okay, that's what we've been talking about. Next, how is that electrical information used to convey information throughout your body? Well, let's think about what it would mean to get a hug. Okay? Think about it. The last time that somebody gave you a nice hug, okay, you feel the pressure of that person giving you a gentle squeeze to the surface of your body, right? And we can have people that give you just a very light hug, and then we can have people that are so excited to see you, they squeeze you and give you a giant bear hug and lift you off the ground. And you can feel, you can understand, you can perceive the difference in the intensity of a light hug versus a bear hug. Well, how in the world is that possible? That's actually a very complicated question to answer, but it all begins with something called a graded potential. Okay, so our next talking point today is the topic of a graded potential. So let's turn the page in our textbook to page 420 and 421. Okay, so I'm planting in your notes that question to think about it while you're studying, considering how you are able to perceive the difference in getting a light hug versus a bear hug. How is this possible? Okay, so if somebody were to give you a hug, and it doesn't matter at this point whether it's a light hug or a bear hug, they're going to actually put their arms around you and squeeze the surface of your body. They're going to put pressure on your skin, right? They are going to literally compress your epidermis and dermis. And you know that you have most of your nerve endings in the dermal layer of the skin. So, okay, so let's talk about this. All right. So, the graded potentials that we did define graded potentials last time. It's in your notes from last time, so hopefully you've had a chance to review that. The graded potentials that generally take place in the dendrites and neurons, they generally take place in the dendrites and cell bodies of the neurons throughout your body. So what I'm saying is, when you get a bear hug when you get a hug of any kind, when somebody squeezes the surface of your body, there are mechanical receptors embedded in the dermal layer of your skin that, because the surface of your skin is being distorted, it's being compressed, it's being manipulated by some external force, those mechanical receptors will in fact open and will allow ions to enter the cell. Okay, if you look at figure 1215, okay, I want you to answer my question through the chat window. Okay. Hold on a second while I get the question typed out. On page 421, figure 1215, is it the top, middle, or bottom photo that shows a receptor that would respond to a bear hug? is it the top, middle, or bottom receptor that would respond to a bear hug? Okay, thank you, Ashley. I know that I can tell that folks are paying attention. That's what I need you to do, right? The top receptor, you notice, shows you that it opens in response to an applied pressure, like getting a hug, like somebody giving you a nice hug because they haven't seen you for a while. Okay, so for graded potentials, okay, most graded potentials occur in the dendrites and cell body of a neuron. Okay, so in our example, if somebody were to give you a hug, these mechanically gated channels would open in response to the pressure being applied to your body. Okay. For a hug, mechanically gated receptors embedded in your dermis would open up in response to the pressure of the hug. Okay. This would allow ions to flow across the plasma membrane of the neuron in the location of the dendrites, okay, or possibly, depending on how the anatomy in the skin is, on the cell body of the dendrite, cell body of the neuron itself. It would depend, okay. All right, and that would change the membrane potential of the neuron. Okay. Okay. So please ask questions if you have them. There's just so much to try to convey here today. So please feel free to ask questions in the chat window. Okay. So typically, when a stimulus like a bear hug occurs, okay, it's typically when a stimulus occurs, it causes a mechanically gated or ligand gated, ligand simply referring to a molecule that can bind to a receptor, a ligand gated channel to open or close. Okay, that's what figure 1215 on page 421 is trying to tell you. Okay, if the stimulus causes the inside surface of the plasma membrane to become more negatively charged, well, let me ask you that through the chat window. If the stimulus causes the inner surface to become even more negatively charged, is the neuron experiencing depolarization or hyperpolarization? What do you say? If this, okay, thank you, Lindsay. Very good, right? If we are making the inside even more hyper negative, then in fact we are experiencing hyperpolarization. If the stimulus causes the inside surface of the plasma membrane to become more negatively charged, the graded potential is hyperpolarizing. Okay. If the stimulus causes the inside to become more negative, then the graded potential is a hyperpolarizing graded potential. And just to complete your notes, If the stimulus causes the inside to become less negative, meaning more positive, then the graded potential is depolarizing. Okay. All right. Okay. So let's see here. Number 3. Okay. The term graded, we're talking about a graded potential. The term graded means that the electrical signals vary in amplitude. Okay, amplitude refers to the size of the electrical signal in terms of its vertical distance. Okay, if you look on page 422, have a look at figure 1216, and you can notice that there's 2 graphs there. The top graph shows you 3 different camel humps, if you will. A green one, a blue one, and a purple one. And you notice that the blue one is higher than the green one, and the purple one is higher than the blue one. Okay, the distance between the tip of the hump and the bottom black line that the hump begins at, that distance is the amplitude. So you can see that each of those different colored camel humps have a different vertical difference, meaning they have a different amplitude, okay? And the amplitude correlates to the strength of the signal, okay? The term graded means that the electrical signals vary in amplitude, which is the size of the vertical distance between the top of the hump and the axis, the x-axis that they are being graphed from. Okay, the term graded means that the electrical signals vary in amplitude. Okay, and this is controlled. The amplitude is controlled by 1, the number of channels that have opened or closed. Okay, so think about it. If somebody gives you a light hug, maybe you have 1,000 channels open. But if somebody gives you a bear hug, they put more pressure, which squeezes more tissue across a bigger surface area of your body. Maybe you have 10,000 channels open up, okay? So the number of channels that have opened or closed is one of the factors that influences the strength of the signal, which is reflected in the amplitude measurement, and how long each channel opens, how long each channel stays open or closed. Okay, so obviously, some channels might stay open for half of a second. Some channels might stay open for 5 seconds, depending on how strong that bear hug really is. Okay, number 4. Okay, graded potentials. They only work over very short distances. That's something we put in your notes from last time. Okay, so graded potentials tend to die out very quickly. All right, that's why, if all we do is produce a graded potential in your skin, your brain's not ever going to be aware that something's happening in the skin. Okay, so what has to happen is the graded potential has to move down the dendrite to the cell body, and if things are strong enough, to the axon hillock of the neuron. And if the graded potentials are strong enough at the axon hillock of the neuron, we will change the nature of the electrical signal from a graded potential to an action potential. Okay, let me put some of this down in your notes here. Because graded potentials act over very short distances, they tend to die out quickly. Okay, because graded potentials act over very short distances, they tend to die out quickly. But their impact, the impact, how significantly you experience an event, how intense the experience is for you, can be influenced by a property known as summation, where you have, rather than one graded potential, you have 1,000 of them, and they're adding their impact into your central nervous system together. Okay? But their impact can be influenced by summation. Okay? And what I'm trying to say here is that this implies that multiple graded potentials occurring near each other can add together to produce a larger effect in your central nervous system. Okay? If we have not just a graded potential at one neuron in the skin, but 500 neurons in the skin, all of that effect can come together to change the intensity of the experience that we perceive in our brain. Multiple graded potentials occurring near each other can add, meaning sum together to produce larger effects. Okay. All right. And you can see that on figure 1217. Okay, look at figure 1217 on page 422. You notice that we have the first stimulus, which creates a graded potential of a certain amplitude, the first part of the purple hump. But then before that first graded potential finishes, a second stimulus comes in the same area of your skin, if you will, during the hug, and a second graded potential is produced, which impacts the overall influence of the graded potentials in that space of your skin, so that the size of the second hump is larger. The amplitude is greater. Okay? Folks, do you have any questions? Okay, let me see here. Pam, so what happens when there are two different graded potentials, one depolarizing and the other hyperpolarizing? Okay, Pam, if you have a depolarizing and hyperpolarizing potentials happening at different areas of the same neuron, they will cancel each other out. If you have some neurons, let's say you had two neurons in your skin that were receiving a depolarizing event, and you had two neurons right next to them that were experiencing hyperpolarizing events, and both of them were connecting to a third neuron down the chain, then they would also cancel each other out. Okay, so generally speaking, if you have opposing depolarizing and hyperpolarizing events, they do tend to neutralize each other. Other questions? Okay, let's see here. let's see here. Okay, so if there's no more questions, we're going to take this idea here and we're now going to transition into the action potential. Okay, so for your brain to become aware of the hug happening at the surface of the skin, graded potentials in the neurons of the skin must be strong enough to activate action potentials in the axons of those same neurons. Those axons can carry action potentials into the spinal cord and up into the brain where you perceive the hug. Okay? For your brain to become aware of the hug happening at the surface of the skin, graded potentials in the neurons of the skin must be strong enough to activate action potentials in the axons of those same neurons. Those axons can then transmit or carry the action potentials into the spinal cord and up into the brain, where your brain can say, oh, somebody's giving me a hug. Okay? All right. Questions before we continue? All right. So let's transition now into a conversation about the action potentials. Okay? This class goes way too fast to get everything explained to you. Okay. So as we said, most graded potentials happen at the dendrites and cell bodies of a neuron. But action potentials typically start in the trigger zone of the axon hillock of a neuron. Okay, so let's see here. If you go back to page 407 of your textbook, okay, 407 of your textbook, all right, figure 12.2. If we stay with the idea of a hug, you're getting a hug from somebody. When somebody presses the surface of your skin, then neurons embedded in the dermal layer of your skin will experience graded potentials at the dendritic spines. You see the dendritic spines. The dendritic spines will have mechanically gated receptors in that open up in response to the pressure you're receiving on your skin from the hug. That will generate graded potentials that move like a wave. You know, if you think about the wave at a football game where people are sitting in the bleachers and one section of bleachers stands up, so the next section of bleachers stands up, so the next section of bleachers stands up. That's the wave I'm talking about. The graded potentials will move along the plasma membrane of the dendritic spines, across the plasma membrane of the cell body, continuing along the plasma membrane towards the axon hillock. Okay, it's at the axon hillock that there's a special section of the plasma membrane that's called the trigger zone, and it's there that the action potential can be stimulated from the graded potentials. So once again, the axon hillock on page 407 is where the action potentials are going to be triggered. Okay, going back to your notes here. Most action potentials start in the axon hillock, specifically the area known as the trigger zone, region of an axon, and then spreads down the length of the axon. Now remember, if we have an action potential happening in an axon that's running down your leg, that axon could be 3 feet long. But it's also possible that the axon is moving 5 inches and making connection with another neuron at your spinal cord. Okay, so the electrical signal of the action potential there would then be converted into the release of neurotransmitter, which would then trigger an action potential in the next neuron, which would then continue the signal, into the brain so that you can understand what's happening here. Okay, let's continue with our notes. Number 2, action potentials. Action potentials are all or none events. Okay, and what that implies is that action potentials are not graded, right? We've just been talking about graded potentials. You can have one graded potential that adds on top of another graded potential that adds on top of another graded potential, and we can get these different amplitude size camel humps. That does not happen with action potentials. All the camel humps with an action potential have exactly the same amplitude. They are not graded, okay? They are not graded, and they have 2 stages, okay? The action potentials have 2 stages, and as you know from your own study time, those are known as depolarization and repolarization events, okay? So let's see here. I suppose page 423, figure 1218 would be a good, figures 1218 and figure 1219 would be good to have open in front of you at this time. So we have depolarization, okay? This is when the inner surface of the plasma membrane becomes more positive, okay, due to opening of voltage-gated sodium channels. Becomes more positive due to the opening of voltage-gated sodium channels, okay? So let's see here. Okay, I will give you more details about that in just a moment, okay? We're going to go into the details of those things in just a moment. And the second phase of an action potential is the repolarizing phase, repolarization. Okay, with repolarization, the inner surface of the plasma membrane becomes more negative again due to the opening of the voltage-gated potassium channels and the simultaneous inactivation of the sodium channels. The inner surface of the plasma membrane becomes more negative due to opening of voltage-gated potassium channels and the simultaneous closing of sodium channels. Okay, so right during your study time, you're going to have to dig deep because you appreciate embedded in the plasma membrane of the neuron, you have sodium and potassium leak channels. That's one category of channel, the leak channels. Then you have other protein channels which also move sodium and potassium, but they're not leak channels, they're voltage-gated channels. So you have voltage-gated channels. And then we also have, never forget, we also have the sodium-potassium ATPase pumps. Okay, questions right now? All right, please ask the questions, and if necessary, turn on your microphone and interrupt me. Okay, so the action potentials are triggered when the minus 70 millivolt resting membrane potential becomes less negative. In other words, more positive. So the action potential at the axon hillock is triggered when the membrane potential goes from minus 70 millivolts to minus 55 millivolts. Okay. If the graded potentials are able to change the membrane potential sufficiently enough so that the membrane at the axon hillock becomes more positive, minus 70 to minus 55 is a more positive change, then you will trigger this all or none event that we call an action potential. And that action potential will propagate like a wave, people at the bleachers of a football stadium, down the length of the axon. Okay. All right, so let me point out that in figure 1219, you notice that you actually have the top picture there, and it shows you the action potential. They have the hump, they have one of the humps being gray, and then they have other humps being blue, but you notice that all of them have the same amplitude, right? They have the same amplitude, meaning they are not graded. Okay, so let me emphasize that in your notes. Unlike graded potentials, the amplitude of action potentials is always the same and does not depend on the stimulus intensity. Okay? The only thing that impacts the stimulus, the only thing that is impacted by the stimulus intensity is the number and intensity of the graded potentials. But if those graded potentials can sum together enough to make the axon hillock more positive inside than it was previously, to go from minus 7 to minus 55 millivolts, we will trigger an action potential, which will propagate unhindered unless there's some disease process going on. Okay. So the stimulus intensity does influence not the amplitude of the action potentials, but in fact the frequency of them. Okay, notice that if we have a really intense stimulus, all the action potentials on the top picture of figure 1219 have the same amplitude, but notice with the blue section there, we have 3 of them together. That's an increase in the frequency of the action potentials. Okay, so the intensity of a stimulus for the action potential is encoded in the number of action potentials in the same period of time. If you have a weak stimulus, you might get 1 action potential a second. If you have a strong stimulus, you might get 1,000 action potentials in that same second. Okay, the stimulus intensity influences the frequency of the action potentials. Okay. So intensity for action potentials is encoded as frequency changes, not amplitude changes. Okay. Frequencies of 10 to 100, 10 to 1,000 action potentials per second are common depending on the strength of the stimulus. Okay. So let's see here. How am I doing on time? All right, not doing too bad, I guess. Okay, Ashley G asks, do these two phases happen one after the other? Ashley, are you talking about depolarization and repolarization? Okay, then the answer to your question is yes. Okay, think about the wave at a football stadium. The first group of bleachers stands up. That's depolarization. The second group of bleachers stands up. That's depolarization in the second group of bleachers. But after the second group of bleachers stands up, what does the first group do? They sit down. That's repolarization. So the action potential will propagate like a wave at the football game in the stadium where the first section of plasma membrane depolarizes. The depolarization of the first section of membrane causes the second section of membrane to depolarize. As the second section of membrane is depolarizing, the first section of membrane will repolarize. So do the two phases happen one after the other? Yes, they do in the same section of membrane. You depolarize first, and then you repolarize afterwards. Yes. Pardon me. Okay. Any other questions? All right. Okay, gang. So I'm trying to look at what I want to cover with you. And so let me talk about the different types of action potential propagations that can in fact occur. Number 5, action potential propagation types. Okay, there's two forms of propagation, if you will. There's a continuous propagation. This occurs in areas of your nervous system that do not have myelin sheath. Continues in areas of the nervous system where axons have no myelin sheath. Okay. So this would be like the wave at a football stadium where every section of bleacher stands up and sits down. Okay. Football game, bleacher analogy. Each section of bleachers, each section of the bleachers stands up and sits down. Okay. But there is another form that, if you're familiar with the anatomy of the nervous system, would be the saltatory conduction variety, which is discontinuous. Okay, saltatory conduction. Okay, this occurs in areas of the nervous system where the axons are wrapped in myelin sheath. Okay. And so, I can modify the football bleacher analogy to accommodate this. Okay, football bleacher analogy. If we're talking about saltatory conduction, then the action potentials are only going to happen at the nodes of Ranvier. Okay, so if you're not familiar what the node of Ranvier actually is, let's see if we can. Yes, back on page 407. Back on page 407, figure 12.2. Have a look at the structure of that axon. You notice the axon itself is colored yellow, but wrapped around various segments of the axon are in fact is in fact myelin sheath, okay, which is made of Schwann cells. Notice that those Schwann cells cover up a significant length of the axon, but there are some exposed gaps. Those exposed gaps are the nodes of Ranvier. Okay, so in saltatory conduction, if you are familiar with the anatomy of the nervous system, the action potentials only occur at the nodes of Ranvier. Okay. For the football game bleacher analogy, what I would say is that for this to work for saltatory conduction, it would mean that instead of each section of the bleacher standing up and sitting down, every other section of bleachers stands up and sits down. Every other section of the bleachers stands up and sits down. Notice the difference between each and every other. Okay. All right. Are you with me, gals? Are you okay? Are you passed out on the sofa yet? Okay. All right. No response. So you must be passed out. All right. Very good. Let's continue here. Just a quick statistic here. Okay, there are different types of nerve fibers in your body, and I'm not going to hit that one too hard, but you'll notice at the bottom of page 426 that your book talks about the classification of nerve fibers. So I'm going to introduce this to you in your notes as I talk about the speed of the electrical signal movement, the speed of the propagation, speed of action potential propagation. Okay, this is going to be dependent on the type of nerve fiber. All right, we have, well, let me do it this way. We have nerve fibers which are called type A nerve fibers. Okay, these transmit to your skeletal muscle tissue, and they can reach speeds of about, well, depending on the location of the body, they can reach speeds anywhere between 27 to 290 miles per hour. That's not a typo. That's correct. Okay. They can transmit action potentials to skeletal muscle tissue at a speed between 27 to 290 miles per hour. Incredibly fast if you think about it. All right. Think about what that means, right? You put your hand on a hot stove and you reflexively use the muscles in your shoulder, back, and arms to move that hand away super fast so you don't burn yourself. That's going to be, that motion, that reflex is going to be carried out by type A nerve fibers. Then we have type B nerve fibers. Okay, these fibers transmit action potentials from organs to the spinal cord. So from your kidneys to the spinal cord, from your liver to the spinal cord, from your heart to the spinal cord. These typically reach a speed of action potential propagation of about 34 miles per hour. Okay, transmit action potentials from organs to the spinal cord at a speed of 34 miles per hour. Also incredibly fast, but not as fast as a type A fiber. not as fast as having to pull your hand off a hot stove. And then there are type 3 fibers, type C, if you will. Okay, they help coordinate your heart rate and control your bladder contraction. I mean, you appreciate that your heart rate, the foundation of the heart rate, is controlled by the anatomy of the heart itself. And you're going to learn that in A&P too. But your nervous system is able to influence the heart rhythm itself. And that's carried out by communication along type C nerve fibers. Okay, these help coordinate heart rate and bladder smooth muscle contraction at a speed of 1 to 4 miles per hour. Okay, which is still pretty fast if you think about the small distance that the signal has to move. Oh, Lord. Okay. Okay. It looks like I'm out of time. So I'm going to stop right here. Lindsay, your question, you said type A transmits the skeletal muscle and type B transmits from the organs. Is the direction significant? Not really. Your book is saying for example, from organs to the spinal cord, but it's bidirectional. It's both ways. If your brain has to communicate to an organ, it will go down the spinal cord and out to the organ. It will go from the spinal cord out to the liver, out to the kidneys. Those are also going to be type B fibers. Okay, other questions? All right. Okay, folks, I'm going to stop sharing my screen. And I'm going to take about a 13-minute break. I will be back online at 9:30. And for anybody that wants to go over the lab exercise homework, I'll be able to do that with you. And if you have any other questions, I'll be happy to do my best to help you, okay? So if you're going to log off, try your best to stay focused. It's still kind of cold outside, so why not take advantage, stay inside with a cup of tea, hot chocolate, and do some really good studying, because before you know it, the final exam is going to be here. Do your best you can. On Monday of next week, I have no choice, but I am going to start the next chapter, which is chapter 15. So we'll be spending all of next week in chapter 15, the following week in chapter 16, and then bang, we are at the final exam. So do your best and let me know how I can help you. Okay, Allie, the next lab exam is May 4th, which is a week from this coming Monday. Okay, anything else? Okay, folks, I'll be back online in about 12 minutes. And if I don't see you, please have a good weekend. And of course, stay safe and do what you can to stay positive, right? Listen to music, exercise, take a walk, do what you can to decrease your stress levels. And I'll see you pretty soon. Take care, folks. Okay, welcome back, everyone. Thanks for sticking around a little bit. Let's go ahead and get your lab homework out, your lab exercises, so we can cover that. If you're following the syllabus tempo, you would have done lab exercises 16 and 17. So let's take a look at lab exercise 16 first. We'll do reviewing your knowledge for that. Okay, so on your homework, page 273 in the lab manual. Reviewing your knowledge, letter A, organization of the nervous system. Okay, so here we have a fill in the blank paragraph. You know, you have experience with that from exams. So let's see how you do here. I'll read the paragraph and put in the answers. Letter A, page 273. If you touch a hot stove with your hand, the sensory, number 1, receptors in your hand send a signal of pain to the central nervous system through number 2, afferent axons of the number 3, peripheral nervous system. When the information reaches the central nervous system and is processed, and number 4, motor response is sent through the number 5, efferent with an E, axons of the peripheral nervous system to skeletal muscles that are number 6, effectors. So the answers from 1 to 6 are 1, receptors, 2, afferent, 3, peripheral, 4, motor, 5, efferent with an E, 6, effectors. Do you need any of those repeated? Okay, letter B. Number 1 and 2 are satellite cells and Schwann cells. Numbers 3 through 6 are astrocyte, ependymal cells, microglia, and oligodendrocytes. 7 through 8 are oligodendrocytes and Schwann cells. 9 through 10 are astrocytes and satellite cells. Turning the page, number 11 is neuron, 12 is ependymal cells, and 13 are microglia. Ladies, do you need any of those repeated from section B? Okay, from section C, the structural classification of neurons. We have number 1, a multipolar neuron. 2, bipolar neuron. 3, unipolar neuron. 4, bipolar neuron. Moving to section D, the functional classification of neurons. Numbers 1 and 2 are motor neuron and interneuron. 3 and 4 are again motor neuron and interneuron. 5 is sensory neuron. 6 is interneuron. Do you have any questions on section C or D that you need repeated? Okay, section E, myelination of the axons in gray and white matter. 1 is B, white matter. 2 is A, gray matter. 3 is C, myelinated fibers. 4 is D, unmyelinated fibers. And 5 is E, the nodes of Ranvier. Okay. Please let me know if you have any further questions on that part. All right, let's go to using your knowledge on page 275, section A, conduction of a nerve impulse. Okay. All right, so we're supposed to reorder the following parts in the correct order of receiving and sending the nerve impulse. Here they are. Number 1 would be the dendrites. Number 2 would be cell body. Number 3 would be axon hillock. Number 4 would be trigger zone. Number 5 is axon. 6 is axon terminal. 7 is synapse. And 8 is the second neuron or effector. Do you need any of that repeated? Number 4. Number 4 is trigger zone. Anything else? Okay, moving to section B, the nervous tissue and the diseases. Okay, identify the nervous tissue cells that are involved in the following diseases. Well, for number 9, with multiple sclerosis, that's typically involving neurons of the brain and spinal cord that have damaged myelin sheath. Okay, so a lot of diseases like multiple sclerosis actually damage the fatty coating around the axon. So 9 is the demyelinated neurons of the brain and spinal cord. And 10 is epilepsy. Well, gang, we really don't know exactly what's happening with epilepsy, but of course the neurons of the brain and how they generate electrical signals in an uncoordinated fashion is involved with epilepsy. So 10 is neurons of the brain. Okay, section C, overview of communication within the nervous system. We are supposed to match the numbers in figure 1610 with descriptions of what is happening at the sensory neurons, interneurons, and motor neurons when they communicate with each other. Start with number 1. Okay. Number 1 would be the statement, graded potential starts in sensory receptor in the skin. Number 2 would be the statement, the graded potential triggers an action potential in a sensory neuron. Number 3 would be the very last statement. At the first synapse, a presynaptic neuron stimulates the postsynaptic neuron to form a graded potential in its cell body, followed by an action potential. Number 4 is the statement. A primary interneuron forms an action potential and crosses the opposite side of the body. Number 5 is the statement. At a second synapse, a graded potential followed by an action potential occurs in a secondary interneuron, which reaches the cerebral cortex. I see your question, Pam. I'll get back to that. Number 6 is the statement. A stimulus from the brain causes a graded potential and then an action potential to form in an upper motor neuron. Number 7 is the statement. In another synapse, the neurotransmitter creates a graded potential, which triggers an action potential in the lower motor neuron. Number 8 is the statement. The lower motor neuron forms a neuromuscular synapse with his hand muscles, which causes the muscles to contract as he writes the letter. So if I'm just reading out the numbers as they refer to the statements that are printed in your textbook, it would be number 1, then number 5, then number 2, then number 8, then number 7, then number 4, then number 6, then number 3. Okay. So Pam, you asked a question about the oligodendrocytes involved in multiple sclerosis. Yes, they are involved, but you know, most of scientific research has involved with the Schwann cells in the peripheral nervous system. But there is evidence to suggest that the myelin sheath that is formed by the oligodendrocytes in the central nervous system are also altered by multiple sclerosis. Okay. Ladies, do you need any of that repeated from using your knowledge or reviewing your knowledge? Okay, let's go to exercise 17. Let's see how you did with that. Exercise 17 begins on page 283. Page 283, reviewing your knowledge, letter A, the meninges. Okay, letter A. Number 1 is the arachnoid mater. 2 is the dura mater. 3 is the epidural space. 4 is the pia mater. 5 is the subarachnoid space. 6 is the denticulate ligament. Denticulate ligament. Do you need any of part A repeated? Number 5 is the subarachnoid space. Anything else? Okay, letter B, spinal cord structures. Number 1 is gray matter. 2 is the posterior median sulcus. 3 is the gray commissure. 4 is the posterior root, which you might also call the dorsal root. 5 is the conus medullaris, the conus medullaris. 6 is the anterior root, which you may have called the ventral root. 7 is the posterior root ganglion. 8 is the cauda equina. 9 is white matter. 10 is the anterior gray horn. 11 is the central canal, where you're going to find cerebrospinal fluid. 12 is the cervical enlargement. 13 is the anterior median fissure. 14 is the filum terminale. 15 is the lumbar enlargement. The lumbar enlargement. Do you need any of that repeated? Okay. All right, let's go on to page 284 there. We have that picture of a cross section of the spinal cord, and it asks you to identify these structures. Okay, page 284. Number 1 is the posterior root ganglion. Number 2 is the posterior root of the spinal nerve. Number 3 is the central canal. Number 4 is the posterior median sulcus. 5 is the posterior white matter that is part of the posterior column. 6 is the posterior gray horn. 7 is the lateral gray horn. Number 8 is the lateral white matter that's part of the lateral column. Okay. Now, on my picture, on my edition of the book, I did not see a number 9. So I think that's an error in the printing of the book. So I'm going to go on to number 10 at this time. Number 10 is the anterior gray horn. Number 11 is anterior white matter in the anterior column. 12 is the anterior median fissure. 13 is the anterior rootlets. 14 is the anterior root of the spinal nerve. And 15 is the spinal nerve. Folks, do you need any of that repeated? Okay, let's go to using your knowledge on page 285. Okay, here again we have a transverse section of the spinal cord. Okay, page 285. Letter A, the spinal cord transverse section. Number one is the subarachnoid space. 2 is the posterior root. 3 is the anterior root. 4 is the dura mater, and actually the arachnoid mater is right there as well. Number 5 is pia mater, and 6 is the epidural space. So, you're going to have a bunch of fatty tissue, adipose tissue in the epidural space. Do you need any of that repeated? Okay, let's turn to page 286, section B on the spinal cord. Number 7 says, identify the area of the spinal cord that is destroyed by the polio virus. Okay, well, the area that is damaged is the anterior horn, Number 8 is asking you about shingles, which is caused by the herpes zoster virus. I see your question, Pam. We'll come back to that. Number 8 is the herpes zoster virus that causes chickenpox and shingles. That virus damages the posterior root ganglion. Number 9 is multipolar neurons. Number 10, talking about the nerve fibers here. In A, we have all three nerve fibers all three nerve fiber types are present in the anterior root. And in the posterior root, we have class A and B fibers. So the anterior root has all three fiber types. Posterior root has class A and B. Number 11. Okay. Explain why spinal taps are not done above the L2 vertebrae. Well, gang, it's simple, right? We don't want the needle to damage the spinal cord. We don't want the needle that's used for the procedure to damage the spinal cord. So that's why we draw the fluid below L2. Okay. So Pam, is your question about using your knowledge section A or reviewing your knowledge? Which part were you asking me about? Using your knowledge, part A, 2, and 3. Okay, let me see here. Will 2 and 3 be the posterior and interior of the spinal nerve? Yes, Pam, that is correct. Okay, let's see here. Let's go to section C on page 286. Letter C is pathways of the sensory and motor impulses. Number 12 would be 2. 13 is 3. 14 is 4. 15 is 5. 16 is 1. Moving on to 17. 17 is 4. 18 is 1. 19 is 2. And 20 is 3. Do you have any part of that section C that you need repeated? Okay, all right. Okay then, folks. So what else can I help you with today? Is there anything else you'd like to talk about? Okay, so Pam, you asked about the scleral venous sinus. You say you cannot find it in the website. Do you mean the website or do you mean the visible body application? Okay, all right. Let's see if we can find that because that seems to be a problem for folks. So let me see if I can bring that anatomy up on my screen for you. Just one moment. Okay, I'm just loading up the software at this time. Let's see. Okay, so I'm going to show the scleral venous sinus and then I will come back to Lindsay's question. Okay, so bear with me a second. I'm going to share my screen. Okay, so at this point you should be seeing the anatomy of the eyeball. And let me bring the chat window up here. Okay. Okay. So we have the eyeball anatomy. Okay. And right now I have the cornea highlighted. I'm going to hide that. And okay. There we go. Notice up here that your software is saying the canal of Schlemm, which is the sinus venous sclera, sclerae, sinus venous venosa sclerae. That's it right there. Okay. At the sclerocorneal junction is an opening known as the scleral venous sinus. Aqueous humor drains into this canal. Okay. So that's how the eyeball drains aqueous humor every day. And that's important to help maintain proper eye pressure. Okay. Does that help, Pam? Okay. So, Lindsay, do you have specific structures you're having trouble with? Because let me say this, everything in your word bank except the spinal cord cross section is findable in the software. So you say mostly the brain structures. Okay, hold on a second. Give me an example of a specific structure that you're struggling with, Lindsay. The pulvinar. All right. Let me, I got to find the electronic version of my word bank here. Hold on a second. Okay, let me print a copy for myself so I can see what I'm doing here. Okay, let's see here. All right, so we have the brain on the screen at this time. And really, looking at these structures, it's just a matter of doing some virtual dissection here, okay? If you're really struggling with this kind of thing, then let me see once, okay? I'm going to suggest that, you do some virtual dissection where we take part of the brain and we get rid of it, right? And then what you want to do is go around and do the internal anatomy of the brain to look for these things, okay? So we have the putamen there, we have the lateral ventricles there, okay? Let's see what happens. Let's see what happens. Okay, if we're really struggling with this, what I would recommend to get your bearings on things is going to Google Images, okay, and typing in pulvinar, okay. So let's see what we can learn from this. Okay. That's not immediately helpful. I know it's in there, so let's see here. All right, let's explore a little bit and see if we can find this, because we should definitely be capable of finding this. Okay, so, all right, it's not the putamen. It's not the cingulate gyrus. It's not the hypothalamus. It's not the corpus callosum. Posterior nucleus, it's not that. Lateral nucleus. Preoptic nucleus. All right. See if we can get some more bearings here. Okay, the pulvinar is the largest nucleus of the thalamus. That's the piece I was missing here. Okay. So, all right. There's the thalamus. Okay, so all right, so we have the globus pallidus. Want to hide that? All right. All right, here's the thalamus. So if we hide this, then what's inside it, we should be able to find the pulvinar. So, okay. So I just hid the outer surface of the thalamus. There it is. There it is. Okay. So the pulvinar is a section of cells inside the thalamus. So you actually have to go inside the thalamus to find it. And really, the ones that you're struggling with, Lindsay, are probably going to be in a similar light. They're probably hidden in some kind of substructure that you need to do some virtual surgery on to figure out what it is. But let's face it, I mean, once you do this, you're going to intimately know where the thalamus is because you had to figure out where the pulvinar was. Okay. All right. So does that help a little bit? Okay. All right. Anything else, gals, that you're struggling with? The spinal plexuses. Okay, is the cervical plexus from C1 to C5. Okay, let's see here. I don't have these things memorized, so let's see what we can figure out together. Okay, well, right away, we figure out that the cervical plexus is from C1 to C4. Okay, from C1 to C4. Now, I don't know off the top of my head, does your software show you that? I mean, it looks like there's 4 connections here. 1, 2, 3, 4. It looks like there's multiple connections there. Don't know why I can't bring up the rest of my anatomy that I want to bring up. But at any rate, yes, it's C1 to C4 for the cervical plexuses, Pam. I would recommend then that you do the other kinds of things on Google. It will help make your life easier. Just do some Google searches and you'll be able to answer your own questions just that much faster. But certainly once you do have the Google searches done, go back to your software and make sure you visualize what that information is so you can see it for yourself. Okay, anything else right now? You know, in your textbook, there are 5 chapters on the nervous system. And honestly, we're only having time to cover 3 of them in lecture and dabble in some basic ideas in lab in the other 2. So even as much as we're doing, we're not quite getting a full comprehensive coverage on this. It's just too deep. All right. Okay, gals, anything else that I can help you with? Okay, Pam, I'm glad you're doing okay. Keep working hard. We'll get through the semester and then you'll get some break. You'll get some rest. Okay, well, I'll stick around, ladies, online. If I can help you, please ask. Otherwise, I'm just going to do a little bit of work here at my desk, okay? But if you need help, by all means, please ask. You too, Lindsay. Have a good weekend.